Hello everyone. Welcome back to MediNotes for Medicals, and the topic that we're going to deal for the day today is intercellular junctions from physiology. So we'll be dealing today with the objectives of introduction, classification, tight junctions, which includes the introduction, mechanism and functions of the tight junctions, anchoring junction that includes cell-to-cell anchoring junctions, cell-to-basal lamina anchoring junctions, and the gap junctions, which includes the connexus and its functions. So what is actually intercellular junction? So if you see over here, you would be seeing that there is a junction, that there are two cells over here and there is a gap between. So this is the junction that is forming between the cells in a tissue, and this gap or this junction where this both connects to each other is known as the intercellular junctions. So now we'll be dealing about the classification. So basically intercellular junctions has been divided broadly into two. One is junctions that tie the cells together, and the other is the junctions that allow the transfer of ions and the small molecules. Now the junctions that tie the cells together are further divided into tight junctions and anchoring junctions. This anchoring junctions is further divided into cell to cell anchoring junctions and cell to basal lamina anchoring junctions. Now cell to cell anchoring junctions have something called zonular adherence, and they have the desmosomes, that is the adhering junction has the protein filaments. These protein filaments are the zonular adherents, or this portion, and there is desmosomes, that is the presence between it, that this is known as the desmosomes. Now if you see about the cell to basal lamina anchoring junctions, you will be having one as the hemidesmosome. So hemidesmosome is known so because it appears like half of the desmosome. So this is the hemidesmosome, this is a complete desmosome. If you cut it into two sections, it forms a hemidesmosome, and it consists of focal adhesions. Now the junctions that allow the transfer of ions and small molecules consist of only one junction, that is the gap junction. Now we will be learning about the tight junctions. So we find certain cells at certain parts of our body. So these tight junctions, they are commonly found in the epithelium of the GI tract, nephrons, urinary tract, hepatobiliary tract, choroid plexus, and the site where they are present in these portions or for example the epithelium of GI tract or the nephrons is at the apical region of this cells, or the apical region of the epithelium of the GI tract, or apical region of the cell of the nephrons. Okay, and the other name of the tight junctions is zona occludens. So they belong, basically these proteins are formed by the families of occludens, claudins, and then the junctional adhesion molecules, and the main function of them are to tie the cells together, and they are selectively leaky. Why? Because they have the presence of ions and water channels. So the mechanism of this is, if you see over here, you would be seeing something called as ridges. So this contributes to the neighboring cells. So at the tight junction, what happens is, if there is another junction, another cell over here consisting of the same ridges, what happens is they come and they fuse over here. So once they fuse, they obliterate to the apical. Okay, so this would be the obliterate to the apical margin, this margin of the intercellular space, that is, they would be binding into this portion over here, and they would be tightly bound to each other. So that is the mechanism of it. And the functions of this tight junctions is that they form selective permeability barrier that does not allow, or it prevents the transfer of macromolecules from the luminal fluid into the interstitial spaces. And they also facilitate the paracellular transport of solute and solvents depending on the osmolality gradient across the epithelium. And then it helps in the permeability of the water soluble particles. How? Via the leaky channels. Then in the ciliary bodies, it acts as a blood aqueous barrier, that is between the inner non-pigmented epithelium cells, and in the brain it acts as a blood-brain barrier. Now we'll be learning about the anchoring junction. So as I said previously, we have the cell-to-cell anchoring junctions and the cell-to-basal lamina anchoring junction. Now the cell-to-cell anchoring junction, they have something called desmosomes. That is, as I showed in the previous slide about the desmosome, it is the junctions that are characterized by the focal thickness of the two adjacent cell membranes. And what happens over here is, in this junction, there are something called cadherin. That is, it is a, if you can see over in the image, this is a cadherin. The cadherin, it is between the two membrane thickening intracellular space, and it also consists of the intermediary filaments from the systole which are attached to this. The systoles would be attached to this thickened areas, and it also has extracellular portions. And now we have the zonal adherens. It is located below the base of the tight junctions, and the major site of the attachment for this intercellular of this is the intermediary filaments. When we go into the cell-to-cell basal lamina, we have anchoring junctions. We have the hemidesmosomes, as I said in the previous slide. It is named so because it appears half of the desmosomes. It consists of microfilaments that are attached to it intracellularly, and as we saw in desmosomes, we have the presence of cadherins. Unlike this, hemidesmosomes, instead of cadherins, it has the presence of integrates. When we go for the focal adhesions are something that that are present at this portions, or it has a main work of connecting the basal lamina to and the cells, and they are associated with active filaments intracellularly. If you see over here, these are the actin filaments, and they assist also in the cell movements. So now we're learning about the gap junction. So we have something called nexus, which is the name that has been given to something between the cells of the narrowed intracellular spaces. Maximally, it is around 25 nanometer in diameter, and it is made up of connexons. Now, what is connexons? Connexons are made up of something called connexins, which are special transmembrane proteins, and they are composed of 6. If you see over here, you can be seeing that there are 6 identical protein subunit combination which are surrounded by aqueous channel, and when it is adjacent, it forms a connection, like it is adjacent to each other and thus forms a connexon, and they connect the cytoplasm of the adjacent cells through their aqueous channel. Okay, now we have the function. So it acts as an electrical synapse. If you see over here, why it acts as an electrical synapse is because the pores of the ligand gate and the voltage gated channels are comparatively smaller in comparison to the gap junction. So what happens is, if the gap is a big, like if the pores are larger, what happens is we have an easy flow of the ions. So once there is an easy flow of the ions, we can easily, that cavity propagations, like rapid propagation, can occur from cell to cell. And the functions of this is, it acts as a physiological symptom also, and there is a passage of organic solutes, for example sugars, amino acids, and we have this exchange of chemical messengers and hormones. It also acts as a regulator. How it acts as a regulator is, it increases the prevention of transport through the gap junction by closing the channels, by regulating the intracellular calcium concentration and the systolic pH. So there is, as you see over here, we have learned about physiological symptom. Similarly, there is something called the functional symptom. What it is present usually in cardiac muscles, visceral smooth muscles. So what it does is, it stimulates one cell. So once the stimulation is done, it activates all the other cells. Why? Because there is an electric impulse that passes through it, and it travels easily via the gap junctions. So that was the video for today. I hope you like this video. If you like this video, don't forget to like, share and subscribe to our channel. We'll be coming with more videos. Still then, happy learning. This is Ashna Viraju signing off for today. Bye.